micro-RNAs are the smallest nucleic acids known. They are just about 20 nucleotides, very, very tiny. They have been discovered relatively recently, in 2001. This year we'll celebrate 10 years, and it really was a huge discovery in biology that sort of really reshaped molecular biology in many ways. They regulate expression of protein-coding genes by regulating protein synthesis of at least 50% of human genes. And by doing so, they play very important roles in normal physiology. They regulate pretty much all cellular functions such as development, growth, cell division, cell metabolism, and many micro-RNAs are often deregulated in human disorders, including brain disorders, and if they deregulated, they may cause or enhance disease progression. So in 2003, it was soon after discovery of this novel class of molecules, I asked myself if micro-RNAs can really have a role in any cancer. We didn't know anything about micro-RNA functions in cancer then, and I was particularly interested in brain tumors. Pretty much then I was a postdoc. Since then I have established a lab, and the focus of our work over all these years was to investigate functions of micro-RNA in brain tumors, and with time it became more translational. We really started to ask questions whether we can imply our knowledge to develop micro-RNA-based drugs and micro-RNA-based diagnostic tools. So let me give you a very brief highlight of our work, because I believe it is important for the project. So in 2003 we developed some tools to study micro-RNAs, micro-RNA expression and function, and these tools appear to be very important, and they led us to identify the first molecule which became first oncogenic micro-RNA called MIR21. We discovered it in gliomas, but it appeared to be very important oncogene in a variety of human cancers, pretty much in all solid tumors. And today, this paper was published in 2005. Now there are more than 300 papers published on MIR21 in all various human cancers. It's certainly one of the most important micro-RNAs in human cancer. By 2008, we convinced ourselves that micro-RNA regulate all various aspects of glioma growth, including ability of tumor cells to divide, migrate, invade through brain parenchyma. Most recently, by 2010, we sort of discovered that micro-RNAs are not only important biologically, but we believe they would represent, they are very important also from a therapeutic perspective. So this slide is supposed to show you that we can inhibit single micro-RNA called MIR10B in established glioma tumors in animal models of glioma. And by inhibiting a single micro-RNA with injections of a small molecule specific drug, we really can reduce tumor growth of established glioma in animals. So all this brings us basically to the current project, and I believe at this stage we really can ask what micro-RNAs would represent the best therapeutic, best molecules to develop therapeutic approaches and to develop drugs for human And the outcome is, we believe that we will know indeed what micro-RNAs are best to be developed into druggable therapies. And another thing is, we believe that the strategy we're trying to validate here should be really applicable to any other tumors for which we have information available, basically. So, for example, we started analyzing meningiomas, and we have published the first paper about micro-RNA profiling in meningioma, and basically a similar approach can be utilized to identify micro-RNAs that might be important for recurrence in meningioma. Basically, this strategy can be pretty much applied to various conditions, various brain tumors. So thank you very much.